Hi everyone, my name is Alexis Thual. I'm a second year PhD student working with Bertrand Thirion and Stanislas Dehaene at INRIA in CEA, France. And today I'll be presenting a piece of open source software that we've been using to compute and explore alignments between individuals using fMRI data and optimal transport. So the problem we're trying to tackle is that of individual variability in fMRI datasets. So I'll jump directly to the piece of software that we use so that you get a better idea of what I'm talking about. So Brain-cockpit is a web-based application that you can actually find online and use yourself. It mainly displays data coming from the individual brain charting dataset, which consists in hundreds of fMRI contrasts acquired for 12 human subjects. And basically here, I'll show you how to display maps for groups of individuals. So here, I'll just select subjects 1, 7, and 9, and we'll display a map, the same map for these individuals. So here, I display a map contrasting areas of the brain activated for math against areas of the brain activated for language. And as you can see, the patterns of activity that we see here differ a lot from one subject to another. Also, in Brain-cockpit, what we can do is look at these patterns on the individual anatomies of these subjects. So bear with me until it loads. But basically what you see here is instead of having this data projected onto a common template, we see the actual image that we acquired in these individuals. And again, what's striking here is that these meshes for each individual differ a lot, so it's kind of hard to compare. So let's go back to the slides. Oops, sorry. Let's go back to the slides. And basically the problem, so we mentioned the individual anatomies, we mentioned projecting them onto a template. And if you take the average of these things onto the template, you basically see that you lose a lot of statistical power, which is kind of the problem here. So what we have is, what we're trying to solve first is to build alignments or couplings, so this matrix P, matching one subject onto another. What we have at our disposal is a lot of features, so a lot of fMRI contrasts that we'll use to build these alignments. And basically the problem that we're trying to solve, so P here will be a matrix of size, the number of vertices of the source subject times the number of vertices of the target subject. And basically the question that we ask is what vertices of the source subject should be matched with vertices of the target subject? And for instance, this vertex I here, we can see its activation pattern throughout the series of experiments that we ran. And we will compare it with the activation patterns of each voxels of the target subject. And we'll try and match things together if they are similar functional patterns. So let's have a look at this in Brain-cockpit. So I'll just open a new window. Here's one subject, and we have a contrast map here that shows areas which are activated for language. So we can click into Broca's area for that specific subject. And so the vertex that we clicked on here, we see how it reacted to each and every stimulus of the data set under study. So basically the big red bars mean that this vertex was particularly activated. The big blue bars mean it was significantly deactivated. And for example, let's focus on this one. I don't know if you can read the name, but so this is one particular contrast, word versus random string. And for that particular contrast, this particular vertex was significantly activated. And so what you can do here is actually, so let's have another subject be put to the side, and we can click on what we think is Broca's area for that subject again. And I don't know if you can see if the resolution is high enough, but basically we just put the two functional fingerprints side by side for these two subjects and these two particular vertices. And this helped me a lot understand how far our alignments need to move on the cortical sheet so as to find counterparts with matching functional fingerprints. So that's basically what we're trying to solve and what Brain-cockpit was useful for in the first place. So let's go back to the slide. I'll just take a minute to introduce you to the optimization problem that we're trying to solve. So let's not spend too much time on this equation, but it's basically a new optimal transport loss that we're trying to minimize here. And basically what we want to find is coupling speed, so these matrices that I introduced before, and we want to find a coupling that will match vertices from the source subject with vertices from the target subject based on the similarity of their functional activity. So that would be that part of the loss. This part of the loss basically tells you that you can't really like shuffle the whole brain altogether. Like you have to, this prevents you from making too drastic changes on the anatomy, on the geometry of the cortical sheet. So for instance, this would prevent you from taking vertices from the prefrontal lobe and matching them with vertices from the occipital lobe, which makes no sense from an anatomical standpoint. I'll just skip the rest, but come and visit us at the poster if you're interested in the rest of the story. But basically what I want to show you here is that the couplings that we derive here, we can actually watch them in Brain-cockpit. So here what you see to the left is the anatomy of the source subject, what you see to the right is the anatomy of the target subject. And we clicked on one specific vertex of the source subject, and we see the probability map of matched vertices onto the target subject. And that's one of the cool things about the alignments that we derive, is that you can control how spread they are. So you can actually decide to compute vertex to vertex alignments, but they can be a little bit more blurry as well, and you will have more spread out probability maps. And so what you can do is actually select another part of the brain, so let's stick in the parietal lobe somewhere around there, and see where it's mapped onto the brain. So as you can see, this one was moved a little to the side, and well, basically that's what we observe in Brain-cockpit. Like we like to see how couplings work and where they send parts from the source subject onto the target subject. You can also do this with individual anatomies, of course, and that's the interesting part. Going back to our problem, I'll just leave you with two ideas about why these couplings are useful and why Brain-cockpit was a useful thing in the first place. So we've seen that if we align subjects with our alignment method, with unbalanced optimal transport, it significantly increases correlation between subjects. So that's what I want to convince you about here. Every dot that you see is one pair of subjects, and so the correlation for these pair of subjects, the correlation of their contrast maps. And so the blue stain that you see here is when we do nothing, so it's just the baseline without alignment. The green part, however, is the correlation once we use alignment. So you see that we significantly gain correlation between pairs of subjects. And the orange part is the current state of the art using a different alignment method. So these results are for the training data set, the training data that we use here, where we see that it generalizes to a test set also for which we acquired different tasks in different MRI sessions. So we're really glad to see that it actually generalizes to things that are not in the training set. The second experiment here is just we change the training set to something a little bit different, and we see that, well, results are kind of the same, but I won't spend too much time talking about this here. Come and see us at the poster. One last thing that I want to discuss here is that this technique is suited to compute barycenters, so some sort of averages of these subjects. So as displayed in the first slide, if you remember, if you take the average of individuals for one given contrast, it blurs out the details that you want to see on the cortical sheet. But if we use our alignment method, so if we align each individual onto a barycenter using our method, we see that we gain a lot of details in doing the averages of these subjects. And basically what I want to leave you with here is the idea that if you compute usual group analysis on just averages, you like areas of the brain which show high variability will just not show up in the t-test that you compute. Whereas if we align brains together in the first place, we see new areas being significantly activated in this setup. So thanks a lot for your attention. I hope to see you at the poster and more generally at OHBM. And yeah, see you.